Okay, so we will move on to the next speaker, Joakim Clifford from Technical University Denmark, also called DTU. He completed his civil engineering degree in bioinformatics and systems biology, during which he gained an interest for solving immunological related problems with machine learning. During his master's thesis, he worked on developing machine learning tools for antibody-agnostic prediction of B-cell epitopes, and then he started his PhD in Morten Nielsen's group with an interest in working on antibody-specific B-cell epitope prediction tools, which I think he will talk today about, also and fusing this with protein language models and structural modeling. Very much looking forward.
Yes,
Yes.
thank you for that nice introduction. And yeah, by the way, that was an amazing talk just before. Yeah, so can you see my screen?
Yes.
Cool. All right, so we'll be talking about predicting antibody-specific targets with structural modeling and epitope-guided spatial restraints. And first, we'll have a quick introduction to B cells and antigens and whatnot, and then we'll get straight into some results. The first is on this paper called, we described this tool called AbEpiTope, which is about assessing the structural accuracy of AlphaFold models. And also, we make a machine learning model that is trained specifically to identify correct antibody-antigen pairing. And then we had this preprint that we just put out, which is about improving structural modeling by predicting B-cell epitope restraints doing Chi-1 modeling. So, as you know, B cells are immune cells which produce antibodies, which they do when they come into contact with an antigen that they are specific for. But more precisely, it's they come into contact with this specific part of the antigen called an epitope. And being able to identify that part and these epitopes is useful when you want to make immunotherapies like vaccines or immunodiagnostic assays. But this identification can be expensive and cumbersome, and so having some like in silico tools which can guide you to sort of reduce some of the experimental workload is quite useful. And there are different ways to go about this. Uh, Probably the one that's been historically most studied is to do it agnostically. And what this means is that you just have an antigen and then you want to predict all likely B-cell epitopes to that antigen, to some generic antibody. Um, and I've worked in one such tool called AbEpiTope. And this is a sequence-based tool. And the idea is that stuff like protein evolution can encode a lot of informative features, like what amino acids are interacting and so forth, essentially almost like structural features. Um, and a couple of years ago, these large language models were coming out showing that a lot of the information that you would have to make a multiple sequence alignment for, you could get that from using a pre-trained language model. And in this paper, we used a pre-trained language model called ESM2 to extract some of that information and gain a performance boost. But one of the problems with these models is that they're hard to evaluate because um, some antigens are just understudied. So we may only have like one solved structure showing in an epitope. And then a model like this will like AbEpiTope may predict other epitopes, but then it's hard to say if these are false positives or, we just don't have a structure for it. And it's only really like prototypical antibody targets like lysozymes, um, or probably like SARS, where we have, the entire surface covered by antibodies. Yeah, so doing that, the specific prediction between an antibody and antigen is more straightforward to evaluate, and it also has more major applications for immunotherapies. As of 2022, there are 162 therapies approved worldwide, and so having some tools which can sort of improve therapy development would be useful. And to model this interaction, we in the first paper use AlphaFold. Specifically, we used AlphaFold 2.3 to make all the structures. And generally, the tasks in this paper can be divided into two. So the first is to assess the quality of AlphaFold structures, or the accuracy of them. And the second is to classify whether or not they were made with the correct antibody and antigen pair. So this is the work uh, or the work process for the first task. First, we have to predict some structures, and our dataset consists of 1,700 complexes from PDB, and we ran AlphaFold with 6 seeds. This means that we generate 30 structures per complex. And then we need to measure the accuracy of all of these structures. And we came up with our own metric, which is called antibody-antigen intersectional union. And this just measures like how well the interface residues between the predicted structure and the solved structure correspond. And it's quite similar to something like fraction of native contacts, which is used in DocQ. But here we also penalize if there are like extra predicted residue contacts. And AlphaFold has its own confidence ranking. And for protein complexes, it's a linear combination of two scores, PTM, which is the overall fold confidence, and AbEpiTope, which is the confidence score between chains. And a confidence score is this linear combination, and this is a score that we have to beat in order for a machine learning model like this to make sense. And the way that we do this is that there's this concept called inverse folding. And it's sort of the opposite of what AlphaFold is. AlphaFold goes from sequence to the coordinates which make up the structure. Um, Inverse folding does the opposite. It tries to predict what residues fit into the backbone atom coordinates of a structure. And ESM1 is this pre-trained one, which was trained using a mask languaging modeling approach on 12 billion AlphaFold and 16,000 CAF structures. And once it's trained on inference, it can give you probability estimates of different regions in the structure, but also like informative embeddings of different regions. And we used these embeddings to featureize the AlphaFold structure antibody-antigen interfaces. So that's getting the labels and getting the features, and now we can train a machine learning model. But first, we have to partition our dataset. Um, so the way we did that was we made 5 data groups, and we ensured that if you looked across the data groups, there was no more than 65% or 95% antigen or antibody sequence identity, respectively. And then once you have these groups, you can then combine them and go through all the combinations to do nested cross-validation. And then it's in the test scores that we use to do all the evaluation and the plots. For training the regression neural network, it was what worked best was actually just to average the embeddings of the interface and then feeding through a very simple regression feed-forward neural network. What was important, however, was to batch structures for the same complex. This made it less noisy and easier for the model to learn a signal. And when then evaluating how well the scores correspond to the actual metric, the accuracy metric, so intersection union, compared to alpha confidence, we get better correspondence linearly. But this is a hexagram plot also illustrating the density of all the structures. Another way to think about it, I've made this illustration where in gray we have a crystallized antibody, in black we have insulin growth factor 2. And what we want is all the antibodies, which are these cartoonish structures, we want all these to be in this area of the plot. So align with the ground truth. And here they're all colored by AbEpi score alpha scores. So we see that AbEpi score is better at scoring structures in the right area better. So that concludes the first part of the stalls. Now we can move on to the next part, which is to identify correct antibody-antigen binding. Okay. Uh, there we go. Yeah, so to do this, we need to create some antigen groups. And we created 700 antigen groups. And One of these is the correct pair, so that's the correct antigen and the correct antibody. And then there are three incorrect pairs, which is the correct antigen, but the incorrect antibody. And these antibodies are something that target different antigens. And a metric we used a lot was this rank 1 accuracy. And this just measures for how many of these antigen groups is the correct antibody-antigen pair the highest. And otherwise, the structure modeling is the same, except now we have to model 3 times as many structures. And also the same featureization. And for the data partitioning, since we did all this swapping with incorrect antibodies, we did that within the data groups from earlier. We could just reuse the same nested cross-validation. So to training the neural network, now we're training a classifier, so with different loss function. Uh, but it's more or less the same architecture. The thing I think is important to say is that we found that batching antigen groups work really well. So the batches for training were we put all the um, like correct antibody antigen pair and all the incorrect pairs together while training the model. If we then evaluate like how well AlphaFold and AbEpiTope are at putting the correct antibody antigen pair ranking at the highest in each antigen group, we get these scores here. And both AlphaFold and AbEpiTope, they can rank the correct pair a lot better than random. But we see that AbEpiTope, which is sort of trained specifically for this task, significantly outperforms AlphaFold. And this is sort of rank 1 accuracy. We also have had made a metric which assesses rank 1, 2, 3, and 4. And this is the score true antibody rank score. And in this graph on the right side, we've sorted all the antigen groups from high to low based on the maximum score in each group, either from a correct pair or an incorrect pair. And then it's AlphaFold or AbEpiTope scores. So in the beginning, we had these groups. All of these in the beginning, there's have a pair which is scoring really highly, and because this pair is something that it's generally the correct pairs which score very highly. In the beginning, we get almost perfect ranking. But then as we go down these antigen groups, the score or ability to identify the correct pair deteriorates. And this delta is sort of a different way of sorting the groups. Here, instead of sorting by the maximum, we sort it by the largest difference between the top two scoring pairs. So the idea is if we have an antigen an antibody pair which scores a lot higher than anything else in the group, that is probably a correct pair. And this does seem to be the case because this dark purple curve displays a lot better ranking ability. Another cool result was that if we look at all the structures and of the incorrect antibody structures, then the correct ones generally receive a lot higher accuracies and scores compared to the incorrect ones. Um, if you look at this upper right corner, there like an incorrect structure cannot really receive this high score or accuracy. And here it is for AbEpiTope, but it is the same if we look at AlphaFold confidence scores. And what this means is that if we can just improve the structural accuracy of AlphaFold predictions, then it's going to make classifying the correct antibody-antigen pair easier. And that sort of concludes this first paper and moves into the next paper where our objective was to improve structural modeling. And since AlphaFold 2.3, there have been a lot of improvements in structural modeling. There's AlphaFold 3, Chai, and Boltz. And in our benchmark, we looked at AlphaFold 3 and Chai, and we looked at how multiple sequence alignment inputs influence predicted structural accuracy. And so on a dataset of 1600 complex, where we use a single seed, meaning we generate 5 structures for each complex, these are the results. And here, Oracle means that we just magically pick the best structure measured by DOQ, which is an accuracy metric. And here we see that AlphaFold 3, if you don't use a template or an MSA, um, performs very poorly, even worse than AlphaFold 2.3, meaning that this MSA input is still very important. But then when you give it the MSA input, it gets this massive jump in performance. And if you look at Chai1, which it performs quite well without an MSA, because in the absence of an MSA, it can rely on ESM, which is a protein language model, but it also gets a large performance bump when you give it an MSA. So all of our MSAs were generated with the MSEEKs, and this dark blue bar plot is just a more sensitive, deeper MSA, and this gave us a small performance bump. So at this point, we were wondering, okay, how do we improve upon this structural modeling? And a cool thing about Chai1 is that it allows you to specify epitope restraints, meaning that if you have some experimental data saying that these are the epitopes, then you can actually give that as features to Chai1. Of course, in many cases, you will not have experimental data showing this. So the idea with this preprint is that we can predict likely B-cell epitopes in the absence of experimental data. And we'll show that this gives a substantial increase in both the accuracy and the diversity of modeled structures. So if you have, the first thing you have to do is make restraints. And if you have a structure, you can use something like Discotope. And if you have a sequence, you can use AbEpiTope. Either way, you can get like this ranked epitope list. And the idea is then you can use these epitopes in consecutive restraint runs, specifying that you want your antibody to be within X angstrom of that epitope. In our run, for the first one, we did not use an epitope restraint, um, just to allow Chai1 to figure it out on itself. This first, yeah. And after that, we ran it with the epitope restraints. And running all these runs will generate like 150 structures for each complex. And to compare, we ran Chai1 as you normally would, just running 30 different random seeds. And what we found is that if we looked at the most confident structures which were being picked, then brown and pink, which are bed pocket and scoop pocket, they can identify a much more accurate structure, or they identify more accurate structures over the entire data set. But what especially like if you look at the oracle, so this is like the total pool of structures which have been made for each complex. Yeah, but the performance is most pronounced for Disco Pocket, which indicates that having a structure-based tool allows you to identify even better epitope restraints. And at this point, we thought that an important factor in this performance boost is probably like how well you model your antigen. So it makes sense that if you cannot model your antigen very well or inconsistently, it doesn't matter how much you look around for an antibody binding site, you will not find anything good. So to test that, we grouped all the complexes by their quality. And it shows that if you look at the poorest group, so these are antigens that have really poor quality, then we also get the smallest performance bump. But then in the highest accuracy group, the performance is almost double this. And we thought that a factor in this performance bump is we must be exploring more diverse antibody binding sites than Chai1 it does when you just give it random seeds. And sure enough, when you look into the data set, you can find these cases where you have this medium to high confidence. And when Chai1 gets this, it sort of just predicts the same learned binding site. But then when you run it with BebPocket, it predicts those same binding sites, but it also explores alternative binding sites. Some are bad, but eventually it does find with high confidence the correct site. And you can quantify that, or we quantify that over the entire data set by making this epitope redundancy. computing this value, and this just measures like if you look at all the structures that are for a complex, like how much do the residues overlap? Um, and this value ranges from 0 to 1, and each dot here is a value for an antibody-antigen complex. And so you can see that the epitopes of structures for when you use web pocket or disc pocket, they're much less redundant. But there is this surprising, like there are actually still some cases which are still quite redundant. And we hypothesize that this is probably because Taiwan is very confident in its prediction, and so it doesn't matter what restraint you give it, it will just sort of ignore it. And to test this, we plotted all the redundancy values against uh, the confidence values. And here we found that there's a positive correlation indicating when you have higher redundancy, um, when it's more confidence predictions, you also get more uh, redundant predictions. But Baby Pocket, Disk Pocket, they are able to like it really reduce the redundancy, especially if you look at this 0.6 to 0.8 area. But then when you get to this above 0.8 confidence, it just seems like the epitope restraints are ignored. Finally, so like Taiwan, Bedpret, and Disco, they all have some training date cutoffs. I mean, we thought that the performance is probably inflated, and so what we did that we split the data set into two. So a complex released after the cutoff and before, and then we did this filtering, ensuring that no complexes had any similarity to stuff in the before data set. And here we found that there's a huge drop in performance, probably like half. But the cool thing is that the performance boost we get from using Web Pocket or Disk Pocket is not really affected by um, this split. So in conclusion, you can predict correct antibody-antigen pairing with AlphaFold structure modeling, and further improvements in structure modeling will likely improve the ability to predict this. And one way of improving structural modeling is to use pocket restraints guided by B-cell epitope prediction. Yes, uh, special thanks to my supervisor, Morten Nielsen, and my colleague, collaborators, and friends for support and discussion. And thank you for listening.
Very nice. Um, could you go back to the conclusion where you say that AlphaFold can be, so how can we understand that we can use AlphaFold for structural modeling of antibody-antigen pairs when um, the evolutionary rules of antibodies are different from general protein-protein interaction? What,
Um,
how
what
do
do
you,
you mean exactly?
I mean,
Yeah, I mean, like, yeah, it's true. Like an MSA doesn't really have any like impact on accurately modeling an antibody.
yeah, yeah, yeah,
Yeah,
yeah,
is that what you mean?
yeah, 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 yeah,
But you can still like you can still model an antibody,
yeah, but
right? Right.
yes, yes.
I mean, a lot of the regions in antibody are sort of conserved, right? So those
Mm.
part you can sort of model, and then it's just a question of modeling like this really difficult part, which is like the complementary determining regions and getting those to fit um, accurately onto the antigen.
Which is the difficult part, right? Yeah.
That's the difficult part. That's sort of like the docking, or and that's why this I think this epitope restraint method is cool because it's sort of like I think it's like really hard for these structure prediction tools to find a nice binding site, and you sort of need to like search the entire antigen surface to find it, right?
Can you explain this epitope restriction or restraint a bit more? How you did?
Yeah,
Yeah, yeah, yeah. Is it?
like the algorithm or this part.
Yeah, I think so. Yeah, yeah,
Maybe go back to this one. Yes, essentially,
yeah.
if you have an antigen where you want to figure out if your antibody of interest interacts with that antigen, uh, then you first score your antigen, and if you have a structure, you'd use a structure-based tool like Discotope,
Mm.
and if you have a sequence, you'd maybe just use BibPrep.
Mm.
But either way, you can like this can give you like a ranked list of antigen residues, which you then can use in like restraint runs,
Mm.
and that sort of corresponds like searching around the antigen
Okay,
for
but
different
but
binding
but
sites.
how? I think I might have asked maybe more than this before, even I don't know. But how does
Yeah.
it work for when you have when an antigen can have multiple epitopes? What does it do then?
Ah,
Yeah.
yeah. So the cool thing about this is that we made like the most simple algorithm to do this. Like we just pick one epitope strain and say that this one epitope has to be within the light and heavy chain.
Mm.
And then it'll try to put it close to that. And then of course there'll be other residues like in the neighborhood, right?
Mm.
On the interaction side.
Mm.
Um, but this is sort of like a baseline, right? I think like if you could come up with more complicated algorithms where you like predict maybe an episode patch or something, and then you use all of that as a restraint, which
Mm.
would
Mm.
probably be more precise.
Mm.
And you could
Mm.
also, I don't know, you could also say that you know that these complemented determining regions should be at the binding site, so you could also give that, um, put that in the restraint.
Mm.
So I think there's a lot of cool optimizations that can be put on top of this.
Yeah, for sure, for sure.
Yeah.
John, you have a question?
Yeah. Hey, great talk. I love the idea. I have a couple of just technical questions. So,
Yeah.
all of your training and sort of your database curation is based on a 95% homology cutoff for
Yeah.
antibodies, right? Um,
Yeah.
so how is that 95% defined? Is it across both heavy and light? Is it just looking at CDRH3?
It's
Um,
either heavy or light.
either heavy
So
or
if we,
okay.
yeah,
Um,
either heavy or light, doesn't matter which. Yes.
Have you ever kind of like looked under the hood a little bit for examples that are working really well and ask what does the
Hmm.
closest like what does that homology cutoff actually look like for this antibody in the PDB or in sequence data? Just because 95% is kind of a low bar, I guess, for antibodies, just anecdotally.
A low bar.
Or however you think about it, it seems like not as stringent as some other work that I've seen, and then the work I've tried to do,
Really?
and because 95% that could mean CDR3 is very similar, but you've just got a couple of mutations that are, kind of in concept, or very similar binders might exist in the training set,
No.
versus it could mean that they're very dissimilar, depending on where that extra 5% is actually distributed.
Hmm. Yeah, I mean,
Especially
yeah,
for light chain, because you can also find binders that are more or less the same, but use different light chains because of the contribution coming from heavy chain.
yeah. I mean, from like clustering, when I clustered the data set, when I did it at 95%, everything gets clustered, almost. Like it's like a completely like a mess. So 95% is not, I wouldn't say it's that low bar. Um, so I think that the process of like making the data sets is like first we figure out if all the different complexes have above this antigen or antibody cutoffs, and then we'd have to like disentangle all the complexes. So um, like remove bottleneck complexes which share identity to many uh, complexes. So
Mhm.
it was like a, a big process to make this data set. Um,
Sure. Yeah.
Yeah,
I mean, and independent of maybe like my bias or whatever, I'd just be interesting to look at examples that are working really well,
yeah,
examples that are not working so well, and actually ask like what are their closest corollaries look like in the training data, just to see if there is any signature that comes out. So we're kind of,
yeah,
without
yeah,
bias, just
yeah.
look
I
at
mean,
that.
it's hard. Like I've only like looked at some examples. Like I
Yeah.
haven't like kind of like looked
Sure, sure,
through my
sure.
entire data set, and then of course I was like you quality check if like other antigens which are in the same in these antigen groups that I made,
Mhm.
which that never I have not seen that happen, right?
Sure.
Um, yeah,
And then
um,
a similar question, but on the antigen side. So for examples that are working very well versus not working so well,
yeah,
because like on the one hand, the sort of hard part is figuring out how to like we think about it on the antibody side because there's less evolutionary information we can leverage on the antibody
Yeah,
side
yeah.
for
There's
how
like
they
nothing.
how the binding is happening.
Yeah,
However,
yeah,
for
yeah,
certain antigens that are super overrepresented in our structural databases,
yeah,
you
yeah,
can
yeah.
think of it about it on the other side where like we just know a lot about how SARS-CoV-2 binds to things, or we know a
Yeah, for
lot
sure,
about
for
how
sure.
hemagglutinin binds to things. So is there sort
Yeah,
of an
yeah,
antigen side bias in terms of examples that are working well versus not?
yeah. I mean, we do like at these complexes, they are redundancy reduced to some extent.
Hmm.
Uh, but there is an overrepresentation of SARS proteins, right? Um, just because like in my data set, I have the SARS complex, the antigen, and then I have like all the different antibodies which have been found to interact with that, and they may, interact with different sites. And so, of course, I want to include all these different sites in my data
Sure.
set. And so, just by that, I have more examples of
Yeah.
that.
It's mostly
Yeah,
like an applicability question. Like for,
yeah,
because, you AlphaFold 3, they say, oh, like we have like a 60% hit rate or whatever. But
yeah,
the hit rate is
yeah.
going to be like
It's
80%
closer
on
to
SARS and it's going to be like
yeah,
10% on some other thing that even though the actual structure of
yeah,
the
yeah,
antigen is modeled perfectly, we just know less about different types of interactions with those proteins.
yeah, for sure.
Yeah. But in any case,
Yeah,
really, really cool work. So thank you.
yeah. Thanks for the questions.
Great. And we are at time. Thanks very much for your talk, Joachim. And thanks again to Dwayne. And thanks for everyone to participate. And we will upload the recordings in the following weeks. Thanks, everyone. Have a good day.
Thank you. See you later.
You too.